All right, so welcome. If you guys want to come closer, you may. I think we're going to only have a few of us. I mean, be comfortable, whatever you want. I'm going to stay here, so if you want to sit there, it's not like I'm going to get up in your business or anything. But my name's Dr. Kate Lysinga-Dean. I am going to be presenting tonight on a topic that's pretty cutting edge, pretty in the news, pretty interesting, especially from a functional medicine, natural medicine standpoint. My colleague, Dr. Stacy, presented on this topic a year and a half ago, and it is astounding how much more we know now than we knew then. It's just going to keep growing too. We'll talk a little bit more about that at the end. There's going to be more and more data coming out. In fact, I get to share with you tonight some hot off the presses data about one of the genetic variants that we're going to talk about. I literally just learned about this a few days ago. So the point is that this seminar will probably be 2.0, but there will be 3.0, 4.0, 5. I mean, we're going to just keep learning. It's just going to keep going and going. So that's exciting. But it does mean that we don't have all of the information tonight. I'm going to tell you a bit about what we do know, with really a focus on what do we do about it, right? Because it's nice to have the information, that is a helpful thing, but it's more nice to know, okay, so what does this mean, right? So that's really my focus today, because you can get all wrapped up in a lot of the nitty-gritty and the details and kind of with your head spinning at the end, but I just want to make sure that we try to keep things very practical tonight. Okay, so that's one of the goals that I have, which is why we're going to talk about this as a beginner's guide. So when we talk about the topic of genetics, there is sort of an inherent identity when it comes to our genes, right? We sort of start to ask the question, who am I? What does this mean? What does all this genetics stuff have to do with me? The me that I know, that I understand, the me that I grew up with, right? The me that is part of my community and part of my family. And that's an interesting way to look at genetics, right? My argument tonight is going to be that it has more to do with nurture than it has to do with nature. That's kind of the push-pull here, right? Is how much of this is the biological makeup of me, the ingredients that make up my body, how those chemicals interact with each other, and my environment, right? My internal and external environment. So one way that I think about that is a lot of us think of our genetics as hereditary, right? So when we think about the heredity of our family, say, maybe we look like our family members, maybe we have the same eye color, maybe we have similar face shapes or body types. Can anyone relate to that? Yeah, some of us look, and as we age, then we think, oh, now I really look like my mom, or, these things start to come out. My funny story with that is when I was in middle school, a very well-meaning member of my church at the time came up to me and she said, you would look just like your dad if you had a beard. And I was like, thank you. Just, the awkward middle school, totally crushed inside. I think I walked away just like if only I had a beard, right? But you know what that's like. You know what that's like to kind of look like our family. Now, on the flip side of that, how many of you have been told that maybe you have the same mannerisms as your family, right? The same way you walk, the way you gesture with your hands, the way you say our words, right? That has less to do with our genetics and a lot more to do with the environment in which we are raised. And I'm going to talk about genetics actually in the same way. We don't necessarily have to view genetics as a fixed point in our lives. They're actually much more flexible than we think, and they have to do with that interaction with our environment. In the same way that our genes don't necessarily tell us who we are, they don't dictate our future like we thought they would. When scientists first started studying genetics, it started with the Human Genome Project. That was like a crazy 13-year, very expensive thing that scientists pooled their resources together, and they just wanted to map out the human genome. And they thought that if they could figure out all of these genes and if they could map all of that out, then we would understand the cure for all these diseases. We would be able to prevent all of these issues. I mean, it would give us like the magic info. We would have the full scoop on who we are as humans. And spoiler alert, that's not how it worked out. We found layers on layers on layers on layers of how our bodies interact with our genetics. And genetic expression is one of those layers. So you've got your genes, and we're going to talk a little bit more about this. But what matters a whole lot more is how your genes are expressed. How our genes create the products of our genes, right? Proteins, and I'm going to explain that all in more detail in a second. But that genetic expression, especially as it relates to our internal and external environment, is a heck of a lot more important than the building blocks or the genetic code that exists in our bodies. And we can intuit that, right? Most of the time, our genetics are not as cut and dry determinative, right? Some people have, the same or very similar genetics and end up in very different paths with their health, with their life, with their personalities, right? And we understand that even twins, right? People who share a ton of the same genetics, if given different situations in life or even similar, one can end up down this path and one can end up down this path. So we have to appreciate that that's a part of our genetics, that genetic expression. The awesome thing about this is that it gives us something to do. It means that our genes are not determining our future. They don't dictate what happens to us in that sort of fatalistic way that maybe we thought a long time ago. So I'm going to talk more about gene expression than I am about your actual genetic makeup, because that's what makes a difference in how our health and our wellness plays out in our lives. Does that make sense? Yeah? So whenever I start teaching on a topic, I want to make sure that we're all speaking the same language, okay? So we're going to go through just a couple terms and definitions so that when I throw these out there later, hopefully it will make a little bit more sense. Genetics is a very technical topic. A lot of us understand the process of digestion. We kind of get the basics of that, right? We kind of understand some of these bodily functions, but our genes, that feels a little bit tricky. So I just want to make sure that we're all on the same page first. So genes are the instructions. I was taught genes are the blueprints. I think that a better idea or a better analogy is that genes are a code because they need to be interpreted, okay? Blueprints are very, right, they're cut and dry. They're the instruction manual. It turns out that our genes are actually a little bit more like a code that you can kind of jumble up and rearrange and create more different outcomes than like a traditional map would be. So I like to think of the genes as a code that we have to interpret in order to get us the information that we need and get us the products that we need. SNPs we call SNPs. That's kind of how the jargon goes. Single nucleotide polymorphisms, which I'm not going to continue to say all night. I am going to say SNP. These are the variations. Now, a lot of people, SNPs get a bad rap with a lot of people. They think of them as errors, okay, in our genetics. But they're not really errors. They can be good things. They can actually convey helpful things, right? So there's a SNP that we're going to talk a little bit about in the FUT2 gene, FUT2, that actually makes you immune to norovirus. Unfortunately, it has a lot of other negative things that go along with that, but there's always a give and take with these variants. So that's why I refer to them as a variant and not as an error or in your genes. Does that make sense? So those are SNPs. Epigenetics are the process by which our genes are translated. Okay, so that's the expression of our genes is epigenetics. So you've got people studying genetics, and then now there's this whole new field of epigenetics that people are studying. And then there's going to be a whole nother layer of microepigenetics, where they study the genetics of the way our bacteria interact with our own genes. Right? This goes, like I said, it's like down the rabbit hole. It goes on and on and on. So epigenetics is that process. Methylation is how we express our genes. So how we change our genes' expression happens by a chemical process called methylation. And some of you have heard that term kind of tossed around, methylation. Oh, it has to do with detox. So I'm going to really get down to the nitty-gritty and explain exactly what we mean by that, especially when it has to do with genetics, because that has a big role. And then proteins are the product. So that's what actually gets made at the end of the day from our genes, is we've got these chemical products. A lot of them are proteins. Not all of them, but a lot of them are. So that's kind of the end point of all of this genetic translation and decoding and expression and turning on and turning off, okay? So when we talk about those things, that's the way that we're gonna go. SNPs, I mentioned this earlier. So the thing about SNPs is that we have 2 copies of a gene. So you get one from your mom and one from your dad, okay, with your genetics. If everything goes right. If it doesn't, then you've got bigger problems, right? But we want to have one from each. And if we are heterozygous for a gene, that means we have one with one SNP and one with a different SNP, okay? So you get one copy of one kind and one copy of another. And I'll explain that a little bit more later. Homozygous means they're the same, okay? So they're either this kind or they're that kind, variation-wise, okay? And that plays out in different ways. A good example is with severity of some genetic disorders. So some people can have a genetic disorder, but it's not as severe as another person. ApoE4 is a genetic disorder that confers Alzheimer's disease, late onset. And if you've got one gene that has the ApoE4 SNP and one that does not, that could explain why some people have a milder form of Alzheimer's than if you have 2 of those negative SNP genes for Alzheimer's. Make sense? Or if you have 2 of the non-SNP genes, then you'd be fine, right? Then there's less of a likelihood that you're going to get Alzheimer's. Make sense? So you've got kind of one from the other. So it's not all or nothing. And if you've got one normal and one abnormal, then you've got a likelihood of a less severe disease, or maybe even not, because one of those genes might be enough to make up for the loss of the other gene. So it might just be a slowed down process too. So when we talk about these, we might talk about whether they're hetero or homozygous, and it helps us understand how much support we need to give these genes or how determinative they might be. For your reference, the RS number is actually assigned to each SNP. So there's an ID number, and it's like a couple letters and a couple numbers long. So if you happen to get some genetic testing done and you get the raw data, you can actually a lot of times search for these if you know the RS number, so that you can find out, oh, do I have heterozygous or homozygous or any SNPs in this area of my genetics? So that's a really useful number if you want to get into the nitty-gritty of some of these later. If you've got a 23andMe, you can Google RS number for FOOT2, right, or something like that, and you can search in your 23andMe results and figure that out. And I'm going to talk about that more in a second. Let's bump back to epigenetics very briefly. I want to walk you through this little diagram because I think it helps us understand a little bit better how genetics works. So you've got your DNA here. So this is your DNA chain, all right? And it's got these little markers on it as we go through, right? And you can see here, this is a methyl group. So this is when I was mentioning methylation. So this methyl group was added on, and that either turns the gene on or turns the gene off, depending on the gene. Okay, so let's say it's a proto-oncogene, so it's a gene that starts cancer so it starts that whole process happening where cells start to rapidly divide. If you've got a proto-oncogene that's turned on by a methyl group, that's not good, right? We don't want that. So it's not necessarily always a good thing to methylate a gene, but most of the time being able to methylate or unmethylate is the important part. So when we talk about people who have errors in their ability to methylate, that can be problematic because they may not be able to turn on a gene that stops cancer, right? Or they may not be able to unmethylate this thing. Yeah, kind of technical, but that's kind of the idea behind methylation. So the methylation comes in, it turns this gene on, and it signals, okay, so gene, do your thing, right? Like let's make some proteins. So what the gene does is it it calls in this DNA calls in some RNA. RNA here links up to this gene and transcribes, basically. So it takes the message from the DNA that was turned on, and it says, okay, I'm going to make a copy of this, and then I'm going to go somewhere else in the cell, and I'm going to make this protein out of it. Okay, so there's a couple layers of what's going on. It's not like DNA just miraculously, oh, now I've got a protein. It takes some of that, some steps, some interpretation. That's why I like to think of it as a code, right? So sometimes there can be issues with this happening, right? Where it doesn't get transcribed correctly, or, there's a lot of different moving parts that could be problematic. But when we talk about DNA methylation, this is really what I want you to understand, is it turns on or turns off these genes that end up making these proteins, or not, depending on if the gene is turned on. Does that kind of make sense? Yeah? Tracking a little bit? Okay. So now that we understand all of the details, right, let's take a big step back and say, why do we care about this, Dr. Kate? Why do I care about SNPs? Why do I care about messenger RNA? Why do I care about heterozygous or homozygous? Well, because they help us to understand a few things. They help us to understand what risk factors we have. We know enough about some of these genetic SNPs now to understand that we're going to have a tendency towards certain issues in our body if we have this problem, if we have this SNP, okay? So knowing what that risk is very, very helpful, because it helps us compensate for those weaknesses, right? If you don't know, let's say you don't know you have ApoE4, right? You don't know that you have an issue with potentially late in life getting Alzheimer's when you're 25. Maybe you won't take some of those steps that you could be taking to prevent that from happening in the future, because there are a lot of them, actually. The more we understand, the more we know there are all these preventive things that we could be doing. But if you don't know, it's pretty unlikely that you're going to be taking those steps. Make sense? So knowing your risk is huge so that you can compensate. It also gives you more motivation to make some of these changes. Yeah? Because even if it's something simple like, what was that study that you can prevent Alzheimer's by flossing regularly, right? There's a ton of data showing that bacteria from our mouth actually end up infecting our brain sort of chronically over time, and that can cause inflammation that causes Alzheimer's. So if you are like me and you probably didn't floss at all for like the first 10 years of your life, despite what I told the dentist, sorry, you might be a little bit more motivated to do something simple like that if you knew, hey, this isn't going to be an issue for you in the future. Yeah? But some of these lifestyle issues are a little bit bigger than that, right? They're not just flossing. They're changing your diet radically. They're making sure you exercise regularly. They're really, really cutting back on stress. Okay, some of these things that require a lot of effort, if you don't know what your genetics are, you're likely going to have a lot less motivation to make those changes. Yeah? So those are three of the big reasons that they're all kind of going together, why it's really important to study your genetics and understand kind of what you're up against. Because these things are modifiable, friends. This is not, I think people were hesitant to know their genetics because they felt powerless to do anything about them, right? Well, it's genetic. I have met patients like that too. Well, obesity runs in my family, so it's not going to do me any good to change my diet. It's genetic, right? That's actually not the case. It might be harder for you, but you can compensate for these things. There's lots of things that we can compensate, can control, right? Genetics felt out of control. Well, it is what it is, but that's actually not true the way we understand genetics. Okay, so I'll talk to you a little bit more about that. How to find your genetic data. Dr. Stacy did go into this a little bit more in her presentation a year and a half ago. That is on YouTube, so if you need more information about any of these companies, widely available. 23andMe has been the easiest that I have found to, number one, get a hold of, and number two, there's a lot of tools you can use to input that data directly, and they can give you reports like Genetic Genie or MTHFR support. Both of these companies take that 23andMe data and help you translate it, right? So they help categorize, so you don't have to be searching for all of those RS numbers, right? They put it all in a report for you. So that's something that, if that's something, say you're a patient here at DVC and you're really interested in that, doing a 23andMe and getting those reports would be a way that we could go through, you and your DVC doctor could go through together and make some, recommendations or take a deeper look at that. There are also a lot of doctors who are starting to do more specific DNA testing where they don't run the whole thing. They'll just run a few tests. I feel like for the cost of doing the 23andMe compared to that cost, it makes sense to just get all of it. The test is pretty good, it's pretty reliable. So, unless you're really, trying to do everything maybe through your insurance and you can get it covered through that, through your doctor, 23andMe is the way to go. Disclaimer, I have no stake in 23andMe, I just find that it's useful. Okay, so there are other ways to get this information too. This is just the way that I know and that I did for myself. So let's talk a little bit more about methylation, because I mentioned methylation in terms of turning on genes and turning off genes, but there's actually a lot of other stuff that goes along with being a poor methylator. Methylation makes our melatonin, so it can have to do with sleep and sleep cycles. There's a lot of data going back and forth between melatonin and cancer as well as Alzheimer's. Some other big chronic diseases, melatonin not being made well can predispose you to. So knowing that you're a poor methylator might be helpful for prevention there. It slows down cancer and it reduces fight or flight neurotransmitters. So for our methylators tend to be our hyperactive, type A, go, 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 go until you crash people. So if you relate to that, you maybe have an issue with methylation. You have an issue with taking those neurotransmitters down, which can be problematic. You need more yoga, you need more mindfulness in order to balance that out, and you might need some other ingredients that your body doesn't readily make because it has a hard time with this methylation process. And we'll go through that in just a little bit more about what you can do if you're a poor methylator. That's actually the top of my list here. I've got a couple common genetic variants that I want to go through. And when we talk about these, I'm also going to give you some ways to optimize these genes. So we're not going to go back and forth. We're actually going to talk through the whole genetic SNP together, understanding what the issues are, and then understanding what you can do to compensate for that if it happens to be something that you see in your genetic profile. Okay? Make sense? So MTHFR, that's our methylation SNP. There are a number of them actually, but that's sort of the category of methylators. Prone to things like addiction because of that neurotransmitter imbalance, their inability to break those down. Fatigue, depression, all of those things kind of go together. They're very compulsive, but they're also prone to heart disease and cancer because of that, proto-oncogene thing where the methylation can turn these genes on and off on the cells. So if you happen to have this MTHFR SNP, right, either heterozygous, homozygous, we don't know which is which. It's probably one of those that it depends on the severity, right? It's really important for you to get extra pre-methylated B vitamins because in order for your body to really utilize B vitamins well, they need to be methylated. So if you're slow at methylating, if you're not that great at it because of your genetics, you might have these kind of useless B vitamins floating around. So I usually recommend, if I know that's the case for you, I recommend taking 5-methyl tetrahydrofolate. So it's a folic acid with a methylator group already on it. It just takes one step out of the equation. It can be utilized by your body a lot easier whether you methylate well or not. So this is our neuro people, this is our detox people, our cancer folks. And then anybody with neurotransmitter imbalances, we love to use this methylated B vitamin. Cool? So that's MTHFR. And like we said, that's a big picture thing, right? Because with methylation, it's a double whammy. Because not only do you have problems with all of these things, but you have problems turning your genes on and off. So it's much bigger, and it almost encompasses a lot of these other things. And that's why there's a couple different categories of it. So you'll find that a lot of people are testing for this and talking about MTHFR and methylation before some of these other ones start to become more popular. I think A couple of our labs that we work with do an MTHFR specific lab test. But again, 23andMe does cover it, and that website, MTHFR support, can tell you more. CompT is another one. I just saw a statistic, 80% of people have an error in this pathway. That's a lot. And they might not have the homozygous SNP, but some sort of SNP in this pathway somewhere. Again, there's a couple genes that code for this. But it makes sense, doesn't it? I mean, with breast cancer rates coming from estrogen dominance, female hormone issues, that's on the rise. Infertility, right, being one of those issues, that's showing itself a lot more. Anxiety and insomnia, I mean, I don't have the statistics, but I know there's an increase in medications for anxiety like crazy. And I think I can't remember the stat. It was something really staggering though, like 1 in 5 people or something crazy are on some sort of neurotransmitter balancing medication. Insomnia, a lot of people struggle with getting good sleep, either having a hard time falling asleep or waking. And that's because this CompT has a big role in breaking down neurotransmitters. So whereas making things is more methylation, breaking them down is a little bit more with the CompT. So breaking down our estrogen when we don't need it anymore, breaking down our excitatory neurotransmitters, so adrenaline, right? If it just stays in your system, of course you're going to be prone to anxiety, to not sleeping well. And if you have this CompT SNP, then it's going to be more likely. Pain as well, so the excitatory, again, if you don't break down those neurotransmitters, then you're going to be saying pain, 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 pain a lot. And panic goes with anxiety, same idea. So I like to use something called SAMe, that's the abbreviation for S-adenosylmethionine. That's a really good calmer for the brain. It gives the ingredients that you need to make things like GABA, which is more of a calming neurotransmitter in the brain. I found that SAMe works really well to counter things like CompT. And then avoiding xenoestrogens or estrogens from your environment, whether that's birth control pills or hormone replacement or even things from plastics. There's a lot of xenoestrogens in plastics. There's also a new category called metalloestrogens. There's a lot of pesticides and chemicals that act like estrogen when they get into our body. And your body doesn't really know the difference. If it binds to that estrogen receptor, it creates the same response, right? So these things can really be very disruptive with our body's ability to handle estrogens. And this goes for men and women, by the way. By the time we reach 65, I think it's 65, men on average have more estrogen than women of their same age. So we're flipping. Women are decreasing in estrogen production internally, and men have accumulated all of this estrogen. And because of all of these things that are like insulin resistance and all this other stuff that's going on in their bodies as they age, their testosterone is being turned into estrogen. So they have the double whammy of more estrogen than they've ever had in their whole life, and low testosterone predisposes them to all sorts of different issues, heart disease and that kind of stuff. So getting rid of or avoiding these estrogens from our environment is huge for people with this genetic SNP. FOOT2, this is the one that I just, FOOT or foot? I don't know. I'm calling it FOOT, FOOT2, because I think it's fun to say. This is one I just learned about. I'm actually going to read a little bit more about this because this is super interesting. FOOT2 has to do with how you secrete your blood type. So it's an antigen thing. And antigens are huge when it comes to the immune system, right? We have to secrete antigens into our mucous membranes in order to mount an immune response to anything. So the people who are homozygous for this SNP are considered non-secretors. So they don't actually secrete the enzyme that they need to into their mucous membranes. And what that does is it keeps them from feeding the happy bacteria that live on those membranes. So you don't make your prebiotics. So these antigens actually feed the probiotics. And so these people have lower amounts of bifidobacteria And 48% of non-secretors have candida overgrowth. So these are our folks that are constantly sick. They have a really hard time getting over it. They've been that way since they were a kid. Leaky gut, so think celiac disease for these folks. Think imbalanced bacteria, right? So a lot of bloating, a lot of inflammatory bowel disease. And then because of the leaky gut, they get a lot of autoimmunity. So, and all of that is if you've got 2 of those SNPs. You simply do not secrete what you need. So what we can do is we can give you those prebiotics in the form of human milk oligosaccharides. And that's in this product, actually, Ultra GI Replenish. I think it's also SBI Protect, if anybody knows that one. We've used that in the past. I think it's a similar idea. But it feeds those bacteria so that they don't keep dying. And they can actually live in your mucous membranes of your gut and in your sinuses, and right, so that those bacteria stay more balanced because it feeds those, it gives them the prebiotics that the bacteria need. So these people can be on probiotics all they want. It's just going right through them, right? Because they don't have the food that they need to stay in those mucous membranes. And they likely have some other overgrowths because it's been this way since they were a kiddo. The other thing is that if this person's mom was a non-secretor, then they did not get any of this good stuff in breast milk, even if they were breastfed, right? So we usually think of breastfeeding as a fix to a lot of these dysbiosis issues with the bacterial imbalance. But if your mom's a non-secretor, you're more likely to be a non-secretor yourself, and you would have never gotten all of these good human milk oligosaccharides in the beginning. Isn't that groovy? It's like I was talking to a patient right after I learned about this, and I just thought, this has got to be her. This has got to be her. And lo and behold, got her genetics, she's a non-secretor. And she was like, of course, it makes so much sense. I've had these issues since I was a kid. None of my siblings did, but it's my genes. And so she now knows she needs to get a ton of prebiotic foods. She needs these human milk oligosaccharides. She needs to compensate for this weakness that she has in her body so that she doesn't continue to get sick over and over again. Crazy, huh? Is that interesting? Yeah, and that was I just learned that this week. Just kind of cool. So you'll see IBS, any gut-related stuff for here, leaky gut, autoimmunity, chronic dysbiosis, gas and bloating, all of that kind of goes together with this food too. Now, I mentioned this earlier, but people who are non-secretors are immune to norovirus for some reason. So it confers a benefit, right? Somewhere along the line, this gene was a good thing to have maybe when the plague was happening back then. Otherwise, this SNP wouldn't have survived, right? Our body knows what it's doing. It just doesn't always change with its environment the way that it needs to. So at some point during a norovirus plague, somebody developed a FUT2 SNP and their body said, yay, I can't get norovirus. But they also then, right, had all of these other issues because we hardly eat any prebiotic foods anymore. You know, back then we probably were, so the body could get enough. Isn't that interesting? So that's how our body, changes over time and responds. ALDH, ADH, and ALL, they all kind of go together, and they're having to do with detoxification. So if you have SNPs in any of these, you may be more intolerant to alcohol. You may have histamine sensitivity. That's a big one right now that we're starting to study, linking that to things like endometriosis, things like leaky gut. We're linking that immune response of histamine to those issues. Chemical sensitivity, because you don't detox well. And cancers, again, because your body doesn't detox well. So we all know detox is super important. Some ways to make sure that your body gets a leg up when it comes to detoxification are sulforaphane. It's from broccoli. So you take one of these little pills, it's like eating a bushel of broccoli. We get all the goodness out of that. Milk thistle is another liver tonic, and N-acetylcysteine makes glutathione, which is a big deal when it comes to detox, being able to detox your body. BRCA1, BRCA2, we know BRCAs, breast cancer, right? Again, this homozygous, heterozygous thing, why do some people get it and some people don't, even though they have the gene? It's your environment. Are you estrogen dominant? Have you been on birth control forever? right? I mean, there's a lot of different things. Are you actively detoxing your xenoestrogens? Are you drinking out of, a plastic bottle, water bottle that's been sitting in the sun? Or are you drinking out of your stainless steel, BPA-free bottle? How much exposure are we getting to plastics or xenoestrogens or phthalates in our products that we put on our face, right? All of this stuff plays a role in the epigenetic layer of things like BRCA1, BRCA2. So you have the risk, then you can step back and say, okay, I need to do all of these different things. Sulforaphane, again, is really good for these folks, avoiding xenoestrogens, hormone replacement therapy avoiding, and then this Axis Endo is a really good one to detox from estrogen. It helps your body get rid of excess inflammatory estrogens that are lingering in your system. So anytime we can do that, that's excellent for breast cancer prevention. ApoE4, I mentioned this earlier, late onset Alzheimer's. We are learning so much about Alzheimer's prevention. I mean, more and more and more coming out because it's such a scourge on our society. It's robbing people of their golden years, and it's one of these things that, we can see now reversal of this. But it's better prevented than treated. Alzheimer's, we do so much better. We have so much power and leverage before we get to a point where we're symptomatic. So trying to convince people to take these steps now is a heck of a lot easier if they know, oh, I'm predisposed. Yeah, so knowing your genetics is helpful for that. We have a wellness essentials pack that covers a lot of these, vitamin D, zinc, and omega fatty acids. We need a lot more fish oil than we thought. We used to think 1 to 2 grams. Now they're saying 3 to 4, maybe even 6 grams a day, which is quite a bit of mixed omegas, especially if we know that this is an issue for us. Hardly any of us get enough in our food. It's just very hard to do. Zinc is a big one connected with Alzheimer's. And then eating a high-fat, low-carbohydrate diet, especially a more ketogenic type diet, they're finding that the folks that do this are the ones that see the most symptom reversal. They're very strict on feeding their brain ketones instead of carbohydrate, glucose molecules. So they put it together in a pack because brain health is pretty essential. So that's an easy one to pick up if you've got any issues with family Alzheimer's, or if you've got this ApoE4, it kind of covers a lot of your bases. So those were the main SNPs that I wanted to talk about, the ones that I have the most data on. And I hope that you could see how practical this was, right? That if we understand these genetics, it's not just our fate, it's not our destiny, it's not who we are, but it actually gives us action points. So number 1, methylate, methylate, methylate. If nothing else, it would be good to know if you've got an error in one of your methylation pathways. That's broadly applicable for everyone, data. It's not a scary one. It's very, you can very much overcome that one. So that would be one thing to make sure that you're doing is methylating well. Assess your risk. We talked about that. Kind of know the score. Make sure you're compensating. Prevention is key. And manage your internal and external environment. So whether that's staying away from plastics, whether that's watching your stress levels, whether that's feeding your brain better, right? Those are the kind of things that we can really change and manage that have a measurable, testable, resultsable effect on how your body expresses your genetics. And this, again, it's the tip of the iceberg. I could do this again in 6 months and have a totally different seminar for you. Maybe I will. Because it's very fascinating. It's applicable to everybody, right? Nobody gets out of the fact that we've got genes. It's not like anybody can be like, well, I don't have genes, so I don't have to worry about that, right? We all do. So knowing what we can change and what we are working with is huge. But optimizing that, making it actionable is the most important part and the take home for this. So as we learn more, we will pass that along to you. And if you come up with anything, if you get your genetic printout and you have questions about it, please ask us. That's something that we study for, we're learning about. We don't have all the answers, but we're actively moving that direction because of its relevance. So that's super important. We could all make some changes, but it's good to have a direction to go, because you can't do everything, right? And so this also gives you a little bit of guidance as to where to focus your energies and, you know, where to look first. So, cool. Well, if you have any other questions, let me know. I'm going to hang out for a minute, but thanks for coming, everyone. Really appreciate it. Thank you.